There's all this talk about self-resynthesis, but what exactly is it? What exactly does it mean to, synthesize a protein using cell-free synthesis?
Right. So it means that we've grown up cells under specific environmental circumstances so that we have the right metabolic capability that we need. We then harvest those cells and we rip them apart, we tear them open, and we take out the insides, and then we activate the insides to produce our protein. And by doing that, we now have direct access to those reactions, which are normally hidden inside of a closed shell inside of the cell. Now we've opened this up so that we can control these reactions much more precisely.
So by not using self-resynthesis, should we really be able to have such direct access into these organisms?
That's right. So to mature these hydrogenases, we need the helper proteins, and we also need a number of small molecules. So we need S-adenosylmethionine. We need a sulfur source, such as sulfite. We need an iron source. Turns out we also need NAD, nicotinamide adenine dinucleotide, and we need a couple other molecules as well. And so because we've opened up the system, we can add those in precise amounts and really control this activation reaction.
What goes into, like coming up with this evolved protein in order to make it more tolerant to oxygen?
Right. So there's some key components that go into that process. The first is to generate genetic diversity.
Okay, and how do you do that?
We typically do that by doing polymerase chain reactions, which are actually tinkered with to make them less accurate.
So they're error-prone?
It's called error-prone PCR. And there are a couple different ways to do that in a biochemical sense, but the end result is pretty much the same, is that we end up with different sequences. And those processes, those procedures can be adjusted to insert a desired number of mutations in each gene. That's the first step. The next step is that we need to be able to make the product that is expressed or encoded by that gene. And we then need to assess the functionality of that product, in this case oxygen tolerance. But in doing so, we need to maintain the connection between that product and the gene that produced it. And so that's the other really essential feature, two features really, the ability to assess have we improved or not, and if we have the ability to recover the gene that produced that improvement.
After you go through these mutations and you find, oh, I think I've created a mutant that has at least a different hydrogenase activity. How do you go through and isolate them?
Well, we have two different ways that we're working on. The slower but more obvious way is to place a single molecule of DNA, a single gene, into a well or into a tube to amplify that up so that we have many copies. And now we can directly transcribe and translate that template to make our product using cell-free protein synthesis without having to do any more purification or any other modifications. And furthermore, we can then assess the activity of that product and the oxygen tolerant of that product without having to open a cell or without having to purify the product.
Right.
So this makes that process much, much easier. And then we also have the gene that produced the product right there in that reaction. And so it's easy to recover the gene if we see a well or a tube that has higher oxygen tolerance. Now the other way is to use an emulsion. And now each little water droplet in that emulsion can become a separate bioreactor. And again, it's using cell-free protein synthesis in that little droplet. And in that droplet also we have a bead. And to that bead is attached the DNA. And also an antibody that will capture the product that's made from that DNA. And so now on each bead, which comes from each water droplet, we have the product and the gene that produced it. And so now if we can measure the attributes, the functionality of that product and pull that away from the other beads, then we can evolve the product. So we can make it work. It will allow us to look at millions of candidates very quickly using the fluorescent activated cell sorter technology.
In this project, Professor Swartz uses a technology which we've talked about in class. Fluorescent activated cell sorting is a process which screens for products of improved mutants based on fluorescence given off. While very helpful for isolating and screening for mutants, this process is not without its challenges. If you could pinpoint, what would you say is the biggest challenge facing you guys in this project so far?
The screening procedures are very complex, involve many steps, and each step has to be just right. Finding the molecule which only fluoresces after the hydrogenase has reduced it. So the hydrogenase is reductase. And so we add this molecule. If the hydrogenase reduces it, then the fluorescence increases dramatically. And now we also need to keep this fluorescent product associated with the same bead. And so that's yet another requirement for this particular emulsion search.
In these 12 minutes, we have learned a great deal about protein synthesis. Professor Swartz is working on a project that will enable organisms to take in sunlight and create hydrogen gas. But first, an oxygen-tolerant hydrogenase must be created. He will accomplish this by undergoing protein evolution, which involves creating genetic diversity using a polymerase chain reaction which has been altered to produce mutations. Then, he will make more of the mutated gene product by introducing it into an organism that can replicate it. And finally, he will assess the functionality of this new mutated gene to see if it has the characteristics he is looking for. Once they find this gene, they will isolate it either by dilution or emulsion, which will then allow them to use fluorescence-activated cell sorting activity, which we've discussed in class. Thank you so much for taking the time. I know you're very busy. I really appreciate this very much. Professor Swartz and his researchers have found efficient ways of screening for their mutated gene, but have yet to find a hydrogenase that has significant improvement in its oxygen tolerance. They are in the latter stages of this project, and Professor Swartz estimated that they only have about a year left. They have the expression and activation of the hydrogenase working, and once they find a hydrogenase that is oxygen tolerant, they will be able to successfully incorporate it into an organism's genome and create a new source of hydrogen gas.